Hello there. Welcome to a fascinating journey into the world of science and medicine. Today, we're exploring CRISPR, a revolutionary tool changing how we detect diseases. CRISPR diagnostics are fast, precise, and accessible, bringing powerful testing to clinics and even homes. This technology marks a new era in medicine, promising earlier detection and better health for all. Let's get started. Learning about CRISPR and new medical technologies is exciting and empowers us all. But remember, this information is for general education only. For any personal health concerns, always consult a qualified medical professional. Enjoy exploring with me, but trust the experts for your care. Now let's continue our journey. Before we dive into CRISPR diagnostics, let's quickly revisit the basics of genetics. DNA is the blueprint of life, written in a four-letter code, A, T, C, and G. This sequence is unique for every organism, like a barcode. Some viruses use RNA instead of DNA as their genetic code. CRISPR diagnostics can rapidly find these unique genetic signatures, revolutionizing how we detect diseases. CRISPR, or clustered regularly interspaced, short palindromic repeats, is nature's own genetic scissors. Discovered in bacteria, it's an ancient immune system that remembers and destroys invading viruses. Scientists realized they could program this system to target any DNA sequence, revolutionizing gene editing and disease detection. CRISPR's precision and power have sparked a new era in biotechnology. Meet SHERLOCK and DETECTR, two powerful CRISPR-based diagnostic tools. SHERLOCK uses Cas9 to detect RNA, perfect for viruses like the flu or SARS-CoV-2. DETECTR uses Cas12a to find DNA, ideal for bacteria or genetic mutations. Both act as molecular detectives, unleashing a signal when they find their target. SHERLOCK for RNA, DETECTR for DNA, precision diagnostics in action. Let's zoom in on how SHERLOCK detects RNA. It starts with a patient sample, saliva, blood, or a nasal swab. Scientists amplify the RNA using isothermal amplification, making the process fast and portable. Next, a guide RNA and Cas13 enzyme are added, programmed to find a specific viral sequence. Cas13 scans the sample, ignoring irrelevant RNA, and locks onto its target. Once bound, Cas13 becomes activated and starts cutting nearby RNA, including special reporter molecules. These reporters have a dye and quencher. When cut, the dye glows. The brightness of the signal reveals how much target RNA was present. Our results can be read by a handheld device or seen as a color change on a paper strip. From sample to answer, SHERLOCK delivers results in under an hour. Now let's meet DETECTR, the DNA detective. DETECTR uses Cas12a and a custom guide RNA to hunt for DNA sequences, perfect for bacteria or genetic mutations. After amplifying the DNA, the Cas12a guide complex is added to the sample. Cas12a scans for its target, ignoring all other DNA. When it finds the match, Cas12a activates and cuts any single-stranded DNA nearby, including special reporter molecules. Cutting the reporter separates a dye from its quencher, causing a visible glow. The intensity of the glow shows how much target DNA is present. Results are read with a fluorescence reader or on a paper strip. DETECTR turns complex DNA searches into clear, rapid answers. Let's compare our two CRISPR detectives. SHERLOCK with Cas13 specializes in RNA, ideal for RNA viruses and gene expression studies. DETECTR, powered by Cas12a, targets DNA, great for bacteria, cancer mutations, and genotyping. SHERLOCK uses RNA reporters. DETECTR uses DNA reporters, matching their enzymes' cutting preference. Both systems use programmable guide RNAs for pinpoint accuracy. Their shared strategy: find a unique genetic target, then amplify the signal for easy detection. They're not competitors, but complementary tools for a wide range of diagnostics. The choice depends on whether the target is RNA or DNA. Together, they offer a flexible, rapid platform for modern molecular diagnostics. CRISPR diagnostics stand out for their unmatched sensitivity and specificity. They can detect tiny amounts of genetic material, crucial for early disease detection. The guide RNA ensures the system locks onto the right target, minimizing false positives. Once activated, the enzyme's collateral cleavage amplifies the signal, making even a single molecule detectable. This is a game changer for catching infections or cancer mutations early, even before symptoms appear. For patients, this means more reliable results and earlier interventions. In outbreaks, it helps identify asymptomatic carriers quickly. High sensitivity and specificity build trust in test results. Negative means negative, positive means action. CRISPR turns diagnostics into a proactive tool for health. Speed is critical in diagnostics, and CRISPR delivers. Traditional tests can take days. CRISPR gives results in under an hour. Isothermal amplification and single temperature reactions make the process fast and simple. This speed is vital for emergencies and rapid decision making. CRISPR's real power is its versatility. Just reprogram the guide RNA for a new target. New tests can be developed quickly for emerging diseases. The technology adapts to agriculture, food safety, and environmental monitoring. It can detect plant pathogens, foodborne bacteria, or water contaminants. CRISPR is a universal detection platform, ready for any challenge. Fast, flexible, and powerful, CRISPR is revolutionizing diagnostics. Perhaps one of the most transformative advantages of CRISPR-based diagnostics is their potential for portability. For decades, highly sensitive molecular testing has been chained to the laboratory. It required large, expensive equipment, a stable power supply, and highly trained personnel to operate it. This has created a significant gap in healthcare access, leaving remote communities, low-resource countries, and field-based settings without the benefits of modern diagnostics. CRISPR technology is poised to bridge this gap. Because the reactions can be designed to work at a single temperature and the results can be visualized with simple methods, the entire system can be miniaturized into a portable, user-friendly format. Imagine a diagnostic test that is as simple and self-contained as a home pregnancy test. This is the ultimate goal for many CRISPR diagnostic platforms. Scientists are developing paper-based tests where the CRISPR components are freeze-dried onto a small strip of paper. To run the test, one would simply add a drop of a patient sample, like saliva or blood. The liquid would rehydrate the components, initiating the reaction. After a short wait, a line or a color change would appear on the paper, indicating a positive or negative result. This format completely eliminates the need for laboratory infrastructure, making powerful molecular diagnostics accessible to anyone, anywhere in the world. This portability has profound implications for global health equity. In many parts of the world, access to a diagnostic lab can mean a journey of many hours or even days. A portable point-of-care CRISPR test could empower local healthcare workers to diagnose diseases like malaria, tuberculosis, or HIV on the spot. This would enable immediate treatment, reduce the loss of patients to follow-up, and allow for effective surveillance to prevent outbreaks from spreading. It puts state-of-the-art medical technology directly into the hands of the people who need it most, democratizing healthcare and saving countless lives in underserved populations. Furthermore, this simplicity and portability also benefit well-resourced healthcare systems. It allows for testing to move out of the central lab and into doctors' offices, pharmacies, and even patients' homes. This decentralization can ease the burden on hospitals, speed up clinical workflows, and empower patients to take a more active role in monitoring their own health. For example, a cancer patient could potentially monitor their response to therapy with a simple, regular test at home. The combination of simplicity and portability is not just a matter of convenience. It represents a fundamental shift in where and how diagnostics are performed, promising a future of more accessible, responsive, and personalized healthcare for everyone. CRISPR diagnostics hold huge promise, but standardization is a major hurdle. With labs using different protocols and reagents, results can vary widely, like chefs baking the same cake with different recipes. Without common standards, it's hard to trust or compare results. Achieving global consistency is essential for CRISPR tests to become reliable, trusted tools in healthcare everywhere. Every new diagnostic test must navigate the rigorous process of clinical validation. It must prove its mettle in real-world clinical practice. Extensive studies using diverse patient samples are essential. Researchers calculate key performance metrics like sensitivity and specificity. The test must be as good as or better than existing methods. This ensures patient safety and clinical effectiveness. Regulatory bodies have stringent requirements for approval. Only by proving accuracy and reliability can new tests be integrated into routine patient care. Even with perfect standardization, cost remains a formidable barrier for CRISPR diagnostics. While CRISPR tests promise affordability, initial development and manufacturing are expensive, impacting patients and healthcare systems. For true global impact, prices must be low enough for widespread adoption, especially in low and middle income countries. As demand grows and production scales, costs will decrease, making CRISPR diagnostics more accessible worldwide. When COVID-19 swept the globe, CRISPR-based diagnostics became a powerful new weapon. Scientists rapidly adapted CRISPR to detect the virus and its variants, enabling fast, precise testing. These breakthroughs led to portable, even at-home tests, decentralizing diagnostics for schools, airports, and clinics. CRISPR's success during the pandemic proved its value as a vital tool for global health preparedness. Humanity has long battled diseases like malaria. CRISPR diagnostics offer new hope, detecting parasites and drug resistance with precision. In cancer, CRISPR aids early detection and personalized treatment, revolutionizing care. This technology promises a future of proactive, precise healthcare. CRISPR diagnostics are transforming how we protect our planet and food supply. From farms to factories, these rapid tests will help detect plant diseases, foodborne pathogens, and environmental threats, often in under an hour. With fast, accurate answers, we can prevent outbreaks, safeguard crops, and monitor ecosystems, creating a healthier, more sustainable world for everyone. We have journeyed through the incredible world of CRISPR diagnostics, from its origins as a bacterial immune system to its role on the front lines of a global pandemic. This technology represents a fundamental shift in our approach to detecting illness. It is a move toward rapid, decentralized, user-friendly diagnostics, promising to reshape healthcare. The power of CRISPR lies in its elegant combination of simplicity and sophistication. It empowers us to act proactively, to intervene before symptoms appear, and to manage health with precision. As we look back on our exploration, it is clear that CRISPR-based diagnostics are a cornerstone of the future of medicine. They are already saving lives by providing rapid answers in critical situations. The journey of this technology is a testament to human curiosity and the power of understanding the natural world. By learning from a humble bacterium's defense system, we have unlocked a capability that will protect and improve human health for generations to come. Looking ahead, the future of CRISPR-based diagnostics is incredibly bright and filled with even more exciting possibilities. We are only just beginning to scratch the surface of what this technology can do. Scientists are working on developing multiplex tests, which would allow a single diagnostic strip to test for dozens of different pathogens or genetic markers simultaneously. Imagine giving a single saliva sample and within minutes being screened for the flu, COVID-19, strep, throat, and other common respiratory illnesses all at once. This would provide a comprehensive health snapshot, enabling doctors to prescribe the perfect treatment immediately, saving time and improving patient outcomes significantly. The integration of CRISPR diagnostics with other technologies like artificial intelligence and digital health platforms will further amplify their impact. Imagine a portable CRISPR device that connects to your smartphone. The device could run a test, and an app could instantly interpret the results, provide information about the finding, connect you with a telehealth doctor for a consultation, and even send a prescription to your local pharmacy. This seamless integration of diagnostics, data analysis, and healthcare delivery will make managing our health more convenient, personalized, and efficient than ever before. It puts the power of a sophisticated laboratory right into the palm of your hand. Furthermore, the core technology itself is constantly evolving. Researchers are discovering new Cas enzymes in nature with different properties, some that may be even faster, more precise, or easier to manufacture than the ones we use today. This constant innovation will continue to drive down costs and improve performance, making the tests even more powerful and accessible. The ultimate goal is to create diagnostics that are so inexpensive and simple to use that they become a routine part of daily life, used for everything from monitoring chronic conditions at home to ensuring the water we drink and the food we eat are safe. In closing, the era of CRISPR diagnostics has truly begun. It promises a future where we are no longer just reacting to disease, but are actively and proactively managing our health with tools that are fast, accurate, and available to everyone. From tackling global pandemics to personalizing cancer care and protecting our planet, the applications are as vast as our imagination. This remarkable technology is not just changing a few medical procedures, it is empowering a global shift toward a healthier, more informed, and more equitable world. The future of disease detection is here, and it is precise, it is powerful, and it is for everyone.